Hi guys, this is our topic 6.2 DNA replication. Before we get into it, let's look at our learning objectives and our essential knowledge. Before we get into the learning objective and the essential knowledge, I want to shift your attention to this section over here where it says suggested skills. If you guys recall at the very beginning of our class, when we started our very first topic, we looked at science practices and pretty much what the various science practices are. Now, for questions on your AP exam from this specific topic, most of the questions, or it is very likely that the questions are going to be centered around visual representation. So that's going to be the focal point of the questions that you be asked for this specific topic. And in answering questions that have visual aids or visual representation, you are going to be required to explain relationships between characteristics of biological models in both theoretical and applied context. And theoretical context is like what you've learned, the general context in which I'm going to be explaining the topics to you. And applied context would be applying that to the specific real life examples that you're given in those stimulus questions. So that's the difference between those two. All right, I'm going to have you guys read the learning objectives and the essential knowledge stuff on your own. So you guys can pause the video, you can read that on your own. But I do want to shift your attention to this statement right here. So every now and then, College Board will add what are called exclusion statements for specific topics. And this topic 6.2 does have an exclusion statement. So it says here, the names of the steps and the particular enzymes involved, excluding DNA polymerase, ligase, RNA polymerase, helicase, and topoisomerase, are beyond the scope of the AP exam. What that means is that you do not have to know all the enzymes that will be mentioned. There are several enzymes that are involved in the DNA replication process that will be mentioned that you don't have to know. And there are some enzymes that I'm not even going to mention at all because I do not want you guys to store this in your brain. You don't need the information. However, here are some exceptions. You must know these ones. So you must know and you must be able to explain um, the use of DNA polymerase, ligase, RNA polymerase, helicase, and topoisomerase. So those are within the scope of the AP Bio exam. So you have to know this. All right, so let's get into our topic. So our topic is DNA replication. When does DNA replication occurs? DNA replication occurs during the S phase of interphase. And S phase is the longest phase, and S phase is divided into three other phases. So remember, we have, and I'm attempting to write on here, G1, we have the S phase, and then we have G2, and this is horrible. I'm gonna have to try to do better with my writing. So those are the phases of the S phase. All right, so the next thing we need to be able to determine is how does DNA replicate? So just a quick backstory, guys. After Watson and Crick and Rosalind Franklin were able to determine what DNA actually looks like, so the double helical structure, Watson and Crick, they built a model of DNA. The next question was, how does DNA make copies of itself? Now, scientists were pretty much competing to propose ideas as to how DNA replicates itself. There were three hypothetical models proposed. That is the conservative model, the semi-conservative model, and the dispersive model. So again, scientists were trying to determine how does DNA, we know that DNA is a double helical structure. Now, how does DNA make more of itself? I don't want to waste your time with every single model. So I'm going to go ahead and I'm going to put an X through the models that are not how DNA replicates. So the DNA does not replicate using the conservative model. And DNA does not replicate using the dispersive model. DNA does replicate using what is called the semi-conservative model. So that is the model that is correct, that one in the middle. And what the semi-conservative model, in which and I'm gonna skip over these. The semi-conservative model is the model in which the parental strand will synthesize. So parental strand in gray will synthesize a daughter strand, and the parental strand and the daughter strand or the newly synthesized strand will form an anti-parallel strand, and the bases, using that complementary base pair rule, the bases will join together by hydrogen bonds. So for each newly synthesized, or whenever DNA duplicates or makes a copy of itself, one strand is always going to be the old or the parental strand, and the other strand is going to be the newly synthesized strand. And that is called the semi-conservative model. And as the name semi-conservative suggests, it conserves one of the parental strand, and there is a newly synthesized strand. I'm not going to waste your time with this. We went over this in class. If you guys are interested, I will share with you guys a video in Canvas explaining the details of the experiment. The experiment is beyond the scope of AP Bio, but it's fun, it's cool if you guys are curious as to how they were able to determine, so who and how they were able to determine that the semi-conservative model is the actual way in which DNA replicates. All right, so shifting gears. So here we are, the meat of the matter. Steps in DNA replication. So at the very end of this presentation, you should be able to explain this picture. So what is being depicted in this picture, you should be able to explain that. So we're going to go through the steps in DNA replication. Step one, DNA replication begins at a place called the origin of replication. So wherever the replication or wherever DNA starts to build that newly synthesized strand, we call that the origin of replication. In order for this origin of replication to be established, there are various proteins that do play a role in determining this replication, this origin of replication. And those proteins are responsible for pretty much opening or unzipping the double helical structure. Wherever the double helical structure is opened, and by open we mean the structure, the two strands are now separated, so the hydrogen bonds that held the bases together are now broken. They're temporarily broken because those hydrogen bonds are easily formed. So they're temporarily broken, and we call that region the replication fork. The next step in the process is helicase, which is an enzyme. And remember that exclusion statement. Helicase is not excluded. So it's one of the enzymes that we should know. So this is within the scope of the AP Bio exam. So helicase is an enzyme. We know this is an enzyme because the word ends with ASE. So helicase is the enzyme that is responsible for unzipping or unwinding the DNA. So it's responsible for pretty much opening up the DNA. And by opening up with the DNA, we mean that it pretty much separates the two strands. And remember, what holds those strands together? Hydrogen bonds between the complementary base pairs. So once that helicase goes in, and helicase pretty much separates or unwinds or unzips the two strands, breaking those hydrogen bonds, we have other proteins that we abbreviate SSBPs, and SSBP stands for single strand binding proteins. These guys are responsible for ensuring that the two strands they stay separated until that replication fork has been completely synthesized. So new strands are pretty much synthesized from that specific replication fork, and then helicase moves on, it continues to unzip, and then these single binding proteins, single strand binding proteins will move to another location, or will move to where the fork has now relocated. Um, Second enzyme, topoisomerase. Topoisomerase is responsible for uncoiling are pretty much stretching out the double helical structure. So it stretches out the DNA so that the helicase can pretty much go in between and break those hydrogen bonds between the bases. So helicase is working by separating the strands, and topoisomerase is pretty much responsible for stretching out the DNA. So it's stretching out the double helical structure so that helicase can go in and helicase can do that unzipping. And I just want to draw a little thing on here. So remember, DNA is pretty much coiled up inside the nucleus. So um, it is not perfectly straight. So it's coiled up inside of the nucleus. Topoisomerase has to straighten that DNA out, and after which the helicase goes in, and helicase is responsible for unzipping or separating the two strands. So topoisomerase is always going to be working ahead of that helicase in the unzipping process. And yeah, that's pretty much what happens. All right, so I'm going to quickly show you what I just explained. So here we have our helicase. So helicase in blue. Helicase, please note that helicase is working in the middle of the DNA, and it's separating the two strands. So remember, helicase is responsible for unwinding or unzipping. These are the SSBPs, and SSBPs are responsible for ensuring that these two strands do not bind together until helicase has done, well, helicase is finished unzipping, and also the newly synthesized strands will come between here. So anti-parallel strands are going to be formed. So SSBPs are responsible for ensuring that the hydrogen bonds, because remember, hydrogen bonds are easily broken, but also they're easily formed. So those proteins have to ensure that these two strands remain separate. And here we go. This is our topoisomerase. Topoisomerase is responsible for uncoiling. So topoisomerase is working ahead of helicase. They're both moving in the same direction. However, the topoisomerase is moving ahead to straighten out the DNA, while the helicase is responsible for pretty much unzipping. All right, so the next step that follows is an enzyme that is called primase. The enzyme primase is responsible for placing RNA segments called primers. So primase is an enzyme, and remember, this is beyond the scope of AP Bio, but it's good to know what these things are. Primase is an enzyme that is responsible for placing down these primers. What are primers? Primers are segments of RNA, and they're chunks of RNA that pretty much contains a maximum of 3 bases. And by 3 bases, I mean 3 nucleotide bases. They place them on the parental strand. And remember what the parental strand is. The parental strand is a strand that is being used as a template. So it's a strand that is being read in order to synthesize that new strand. So it places the primer on the parental strand, and once the primer is placed, that is when another enzyme called DNA polymerase 3 goes in and starts the building. Key things from this slide before you move on. You need to know what primers are. That's very important. You also need to know primers are made up of RNA bases. So we're expecting for them to have uracil, and they're 3 base long, or it means that they contain 3 bases. Primers are very important because primers is an indication as to where that replication should start, or where the DNA polymerase 3 should start building. And we're going to talk about DNA polymerase in our next slide. All right, here we go. Before we move on, so just a visual representation. Here we have our primer. The RNA primer is 3 bases long. So 1, 2, 3. This is our parental strand of DNA, and a primer is placed on this parental strand. The primer would have antiparallel and complementary base pairs for these 3 DNA nucleotide bases. However, in the language of RNA, because remember these are RNA. And our fourth step is antiparallel elongation. In antiparallel elongation, as the name suggests, elongation, it is now building that complementary strand. So using the parental strands as templates, it is now each parental strand is being used to build a complementary strand. So what is doing the building? We have our Bob the Builder that is responsible for building these strands, and our Bob the Builder is our DNA polymerase 3. The DNA polymerase 3, it attaches to a primer, and the primers are an indication as to where the DNA polymerase should start building. So you can think of primers as that landing pad. So it's a signal, say, hey, this is where you should start building. And primers are in the language of RNA. So primers are 3 nucleotide based fragments, and they're in the language of RNA, which means that it does have uracil, or it has the basis for RNA. So um, the DNA polymerase, or Bob the Builder, will start building, or it will start elongating the parental strand, and it's going to be moving, or it moves in the 3 to 5 direction. Another way to think of this is, even though it is moving, so it is actually physically moving along the strand. However, I want you to also visualize, or just to have the concept knowing that it is reading is reading in the direction 3 to 5. So think of DNA polymerase 3 reading that parental strand in the 3 to 5 prime direction. And if it is reading the basis on the parental strand, it is pretty much transcribing. So based on what it is reading, it is now placing down these anti-parallel strand or bases. So it is pretty much finding the complementary base pairs for these parental strand, and it's laying those base pairs down in the direction of 5 to 3. DNA, RNA, genetic material should always be synthesized in the 5 to 3 prime direction, and I'm going to get to that in my next slide. So as it is moving or reading in the 3 to 5 direction, it is always going to be building the DNA in the 5 to 3 prime direction. And that's how DNA should always be built. So DNA should always be built, and RNA as well, should always be built in the 5 to 3 prime direction. So please always bear that in mind. The DNA polymerase 3 that follows the helicase. So remember, helicase is going ahead and it's doing that unzipping or the unwinding. The DNA polymerase on the parental strand that is trailing behind that helicase is known as the leading strand. So it is following that helicase. As helicase opens the replication fork, it is going behind and it is building that newly synthesized strand. It only requires that first primer, so that one primer is the only primer that is needed in order for that DNA polymerase 3 to go ahead and start building. This strand is known as the leading strand. Leading strands, they build continuously. They don't stop until it's time to terminate, and they will continue building, and they only require one primer, and they're always going to be going in the same direction as the helicase. Now, on the other parental strand, that strand that is running 5 to 3, or that strand that will be read 5 to 3, there are some issues on that strand. So what that means is that, and I'm sorry, probably not the best choice of words, but what it means is that there is not going to be a smooth, continuous building of the newly synthesized strand on that parental strand that is running 5 to 3, because DNA cannot synthesize the new strand in the 5 to 3, sorry, in the 3 to 5 direction, and therefore we're going to have fragments, or we're going to have lagging strands. So that strand is going to be going the opposite direction of helicase, and it's going to be lagging behind, and it requires many primers. So I'm just circling or highlighting some very important things here that we need to know. Moving away from helicase, lagging behind, requires many primers. Um, those are the key things for our lagging strand. Now I'm going to go to the next slide, and I'm going to try my very best to draw this entire thing that I just explained to you. All right, guys, so I'm going to try to use this diagram right here to explain what was said on the previous slide. So we're going to try to determine how many primers are used for each parental strand, which strand is our lagging strand, which strand is our leading strand, what direction is the helicase moving in, and what direction are these strands being synthesized in. So some key things I want us to highlight here. So let me grab my highlighter and I'm going to choose the color. Let me choose a bright color. So I'm going to choose the color green. Um, this is a parental strand. So this is a parental strand, and this strand right here is also a parental strand. These parental strands are being used as templates. And these parental strands are going to be used to replicate or to pretty much synthesize new strands. The new strands are going to be synthesized here. So we're building a new strand. Now, next thing we need to identify is what strands are considered to be or strand that is running 3 to 5 and what strand is considered to be the strand that is running 5 to 3. All right, so key things here. This strand right here is running 3 to 5. So this is our 3 prime end. So this is our 3 prime end, and this is our 5 prime end right here. So this strand runs 3 to 5. This strand is anti-parallel to this strand, so therefore it is running in the opposite direction. It is running 5 to 3. DNA can only be synthesized in the 5 prime to 3 prime direction. So this is how DNA should be built, in that direction. Now, if we're using a parental strand as a template to synthesize a new strand that must be built 5 to 3, the easiest way to do this is to go ahead and use that strand that is running anti-parallel to 5 to 3, which is a 3 to 5 prime strand. So this parental strand requires only one primer. See primer right here. It uses this one primer. Once this one primer is placed, remember primers indicates to the DNA polymerase where they need to start synthesizing. So primer will allow for the DNA polymerase 3 to read along in the 3 to 5 direction. So it is moving. So our DNA polymerase is moving and it's going to be reading in the 3 to 5 direction. So it's going to be reading this base, and then it's going to be finding the complementary base, and it goes to this base and it finds the complementary base, and it goes to this base and it finds the complementary base, and it's going to be building and building along. And if you see the direction in which I'm building, so I'm acting as the DNA polymerase 3, I'm building in the direction that helicase is moving, because helicase is unwinding in this direction. So I am following behind that helicase, and I'm reading 3 to 5 and I'm building 5 to 3. How do you know I'm building 5 to 3? Because this strand that I'm building is anti-parallel, and if the parental strand that I'm reading or using to build my template is already running 3 to 5, then it means that I'm building a strand that is running in the opposite direction, which is 5 to 3. So as I'm here building this anti-parallel strand, that anti-parallel strand must be running in the opposite direction of the parental strand, which is 5 to 3. No problems here. The only thing I need is just one primer. As polymerase, I've identified where that primer is, and I go ahead and I start building, and I'm trailing behind helicase. And as helicase opens the replication fork up further, I will be building behind that helicase. I am building a long continuous strand, and I will continue building until helicase stops and it's time to terminate. Now, let's shift our attention to the other parental strand, which is this strand that is running 5 to 3. Now, this strand is already running 5 to 3. And remember, DNA must be built 5 to 3. So here I have a problem, because if I should build in this direction following the helicase, if I'm building in this direction following the helicase, it means that I'm going to be building it an anti-parallel strand that is going to be running 3 to 5, right? Now, DNA cannot be built to 3 to 5. So I'm going to go ahead and I'm going to erase that. No. DNA cannot be built 3 to 5. So therefore, what happens with this strand right here, it requires more than one primer. So a primer is going to be placed at the origin where the replication fork opens up. So this is primer 1. And then as helicase is moving ahead, so here again, here's our helicase. This strand, if I'm DNA polymerase trying to build a complementary strand using the 5 to 3 prime, I'm going to have to wait a little bit until helicase opens up the replication fork enough for another primer to be placed. And then once I'm waiting, then I'm going to start building. However, I have to build 5 to 3. So therefore, I have to be building in between primers. I have to build in this direction. So I'm going to have to put a base, a nucleotide base right here. And then I go here. And once I'm doing this, it means that I'm building 5, because this is the 5 prime end. And I'm building it in the direction of the 3 prime end. So I'm building 5 to 3, and I'm building in this direction. And I'm building, if you look, I'm building between these primers. Now, as helicase is opening up more, I'm now lagging behind. So I am called the lagging strand. As I'm lagging behind, I now have to wait. So as helicase opens up some more, I'm now waiting for this primer, and I'm going to change colors here because I probably just confused you. So I'm waiting for this primer so that this primer can jump to another part. So we're going to pretend as if helicase opened it up a little bit more, and then this primer is going to be jumping, and it's going to, let me use red again. The primer is going to go, say, for example, here. Now, once the primer goes here, I have to wait again, and I have to build in between these two primers. So I'm going to be building again in this direction. 5 to 3. And I keep building in between primers. And what I have is I don't have a long continuous strand. Instead, I have fragments of DNA because I'm building in between primers. And I'm sorry if that was confusing. Let me erase this, and I'm going to draw it in the opposite direction. So what I'm doing is I'm building in that direction, but I'm building fragments between these primers. So those are my fragments, and these fragments are called Okazaki fragments, and fragments will exist on that strand, that parental strand that is running 5 to 3, because that parental strand only allows for building in the 3 to 5 direction, and in order to achieve that 5 to 3, it has to wait. So there has to be gaps building in between primers. So that is how that is achieved. I do have some animation on here, which is much better than the scribbling that I just did. So here we have our DNA polymerase 3. DNA polymerase 3 is building, and it follows the helicase on the strand that is running 3 to 5. Continuous building, leading strand. However, my lagging strand is created once the parental strand is running 5 to 3, and polymerase on that strand has to build in the opposite direction. All right, and therefore I have fragments. So it's going to be moving in the opposite direction of the helicase. And I hope that was helpful. And it's going to be building in the opposite direction. All right, so what do we call these? I've mentioned this before. We have on the lagging strand, and you should know what the lagging strand is. On the lagging strand, these fragments or chunks are called Okazaki fragments. So we need to know what these things are, and we need to know how these Okazaki fragments are created. And we're going to mention Okazaki fragments again at the very end, because these fragments they must be glued together by an enzyme that is called ligase. So we're going to mention these Okazaki fragments. All right, so here we have our leading strand. See, continuous building. And then here we have our lagging strand. Look at the direction in which the lagging strand is building. Opposite to the direction that helicase is moving, and opposite to the direction that, what is this guy? Topoisomerase is moving. And that's what creates that lagging strand, and it's being built between primers. And I'm sorry if this is repetitious, guys, but this is pretty much the important stuff that you guys need to know. We're almost there. We're almost there. All right. So, what happens next? So we have another Bob the Builder, and remember, Bob the Builder is DNA polymerase. So after the DNA polymerase 3 forms these Okazaki fragments, there's another polymerase called DNA polymerase 1, and DNA polymerase 1 plays a very crucial role because remember we talked about RNA nucleotides that make up or primers. Now we're trying to synthesize DNA, so why would we need fragments of RNA? So what will happen is these little gaps that were created as a result of these primers, these gaps must be removed, and these gaps are going to be removed to pretty much form a continuous synthesized strand with no gaps by pretty much well, it's simply converting the RNA nucleotides into DNA nucleotides, and it's not very hard because I mean it's nucleotides. The only thing that makes them different is the uracil. So it's switching out the RNA primers or the nucleotides from the RNA primers to make those DNA. And the DNA ligase is pretty much responsible for piecing together those fragments. So those Okazaki fragments are going to be piecing together, and pretty much wherever that primer created that gap so the building could occur will also be piecing together. And here we have our DNA polymerase 1. See, converted that primer into DNA. So there is no gap on that leading strand. And look, voila, it did the same thing for the lagging strand. All right, putting it all together. I think I already did share this YouTube video. If I did not, I'm gonna share it with you guys. So you should be able to look, pause the video and try to explain this to yourself. And there's some multiple choice questions on here, and I want you guys to pause the video, look at the questions, and then try to determine what the answer is. All right, so let's talk a little bit about telomeres. Telomeres are like caps that are placed at the end of the newly synthesized DNA, and that is to prevent the DNA from eroding or the DNA from becoming damaged. So I'm going to go ahead and I'm going to attempt to draw. I'm going to draw a synthesized strand of DNA. So we're going to use two colors. We're going to use green and we're going to use red. Green is like the internal region or the portion of the DNA. I'm sorry, I wanted to make this continuous. There we go. Good enough. So the green region, so I want you to think of the green region as this is actual genetic information, useful genetic information. This is actual genes that can be used to synthesize real proteins. The red region right here is the end in which these are nucleotide bases. However, these are like nonsensical bases. They code for absolutely nothing. Now, the red region right here is repeated nucleotide sequences that protects the green region from erosion. So every time replication occurs, and especially on that lagging strand, because the lagging strand cannot be synthesized to the very end, because wherever it terminates, remember, there's going to be, it's building between primers, so there's going to be a primer at the end. So it means that those genes run the risk of being cut off every time are being shortened. Now, in order to mitigate this or in order to prevent this from occurring, this mechanism is in place in which telomeres or caps are placed at the end of this newly synthesized strand so as to protect the genetic information, which is the green region, from erosion. And I'm going to go ahead and I'm going to erase this little diagram off here, and now I'm going to put some notes on here. And if you guys have any questions about this, feel free to ask during class, or you can watch a video on this. So here we go. These are called telomeres, and telomeres, they do prevent the erosion of genetic information. So genetic information is the integrity of your genetic information is somehow protected, just in case during replication, every time, and replication occurs very often, especially like skin cells that are constantly undergoing mitotic cell division. And every time replication occurs, our genetic information runs the risk of getting shorter and shorter, especially on that lagging strand because it's building in between primers. So it prevents the shortening of the genes, and if they do get shorter, then that green region that I drew, the vital parts are still somewhat internal and they're protected because of that cap at the very end. Guys, we're almost to the end of the lecture. Here's something else that is noteworthy. DNA polymerase can proofread. So DNA polymerase, as it's building the newly synthesized strand, is also checking for errors. So it's also checking to ensure that it's placing the correct bases, the correct complementary bases as it's building that anti-parallel strand. And if there is a base that is incorrect, then it can correct itself. So other enzymes can remove incorrect bases and replace it with the correct bases. And also, if there is a segment of the DNA that is damaged, nuclease can get rid of that damaged section. So that damaged region can be entirely removed by the nuclease. And then another enzyme, which we've mentioned earlier, ligase, can now piece in everything together. So proofreading is also done by the DNA polymerase. However, we know that proofreading doesn't always detect errors because mutation still does occur, but it helps to limit the amount of mutation or the amount of damage to the genetic information. All right, we are at the very end, guys. This is the end of this topic. I know this is a long video. I'm trying to make my other videos a little bit shorter. I'm not an expert at making videos, so this is like my first attempt to actually create a video, guys. So I'm gonna work on it though. I'm gonna work on it, and then when I see you guys again, just give me some feedback. Let me know if this was useful or if it helps, or what are some things that I can improve, because I know that you guys have been watching videos forever, so you guys can give me some tips. Here are some questions and answers to these questions, and I hope as I was on this slide you guys were pausing and actually going through the questions. All right, so I don't want them to make this video any longer than it needs to be, so I'll talk to you guys in class. We're going to do some practice questions, multiple choice, free response questions on this specific topic, and if you guys have any concerns, if there's anything that you guys want clarification on, please ask. And as you guys are going through the videos, I hope that you guys are pretty much taking notes and just ask questions. And if there's anything that you guys want me to add, be a little more thorough with explaining, please let me know.